welcome everyone, and thank you all for virtually joining us for the latest iteration of the Variant Effects Seminar Series, or VES. My name is Diego Calderon, and I'm joined by Stephen Irwood, who is the other member of the VES organizing committee. These seminars are made possible with support and a lot of help from Laura Muffley, among several others, and our sponsor, the Atlas of Variant Effects Alliance. So a big thank you to them. Before jumping in, I have a few housekeeping items to mention. Each talk is about 20 to 25 minutes, so there should be time for a few questions. Please post any of those that you might have to the Zoom Q&A or chat, and we'll read them at the end. And the presenters should feel free to respond as they like. If you have additional questions after the end of the seminar and you're an AVE member, feel free to visit our seminar series Slack channel for additional discussion. You can catch the seminar today and previous presentations that have been recorded on our CMAP YouTube channel. And finally, we're now posting updates on our Twitter account, so be sure to follow us there at Variant Effects. This has never been an issue, but as a reminder, please be considerate when posting comments or questions. We expect everyone to be respectful and use welcoming and inclusive language. All the details of this seminar series can be found on our website. I just wanted to quickly notify everyone that we're going to skip the January seminar, but we'll post an update shortly for information on our February series. And without further ado, I'm very excited for today's presentations. First off, we have Mireia Seuma presenting. Mireia is a PhD student in Benedetta Bolognesi's lab at the Institute for Bioengineering of Catalunya in Barcelona. In their lab, they use deep mutational scanning to understand and predict how genetic variation impact amyloid formation in Alzheimer's and other neurodegenerative diseases. The title of her talk is Mapping Amyloid Nucleation by Deep Mutational Scanning. And Mireia, feel free to start sharing your screen and take it away when you're ready.
Thank you, Diego. So I hope this works now and you can hear me well. Right. So, well, hi, everybody. As Diego was saying, I'm Mireia Seuma, a PhD student at Benedetta Bolognesi's lab at the Institute for Bioengineering of Catalunya in Barcelona. And today I'm very happy to be here in this nice seminar series and to be presenting our recent project in which we mapped amyloid nucleation by deep mutational scanning. And to start with, I want to explain a bit what we are doing in our lab. So our lab is interested in those proteins that can adopt different conformations and undergo a process of self-assembly. And this is the case of the amyloid beta peptide, which is the main component of extracellular amyloid deposits in the brain of Alzheimer's disease patients. And the formation of amyloid fibrils is controlled by nucleation. Then what does this mean? This means that for fibrils to grow, you need to first form a critical assembly, and this has to reach and actually surpass a high kinetic barrier to then seed further aggregation into amyloid fibrils. And you have to keep in mind that this is an irreversible process, and so nucleation is the critical step. Once it starts, there is no way back. In this peptide, there are 14 mutations that are associated to inherited forms of Alzheimer's disease. And there have been some in vitro studies for the disease variants and a few other variants. You see the ones that are associated to disease here in red. And some of them are known to increase aggregation. Some of them are known to decrease aggregation. But overall, there is no huge consensus on their effect on nucleation. And actually, there's not even one study in which all 14 disease mutations have been tested together. And so here we asked, I have to say that all other variants in this peptide are of unknown clinical significance. And here we asked ourselves, how do all possible mutations in A beta alter the propensity, all mutations alter the propensity of A-beta to nucleate amyloids. And to answer this question, we developed an assay that, coupled to deep mutational scanning, reports on amyloid nucleation. So this is our assay, and we first built a library of more than 14,000 single and double amino acid substitutions that we introduced in a Jesus strain with a premature stop codon in the adenine gene. Our library is fused to the nucleation domain of sub35, which is a well-known GIS prion and a translation termination factor that gives a phenotype upon conformational change. So if a specific variant of A-beta here nucleates, it causes the nucleation of sub35 that will recruit endogenous full-length sub35 protein. And once aggregated, sub35 cannot do its job anymore. There's a read-through of the stop codon, and this cell can now grow in a medium that is lacking adenine. And we have shown that this selection assay directly relates amyloid nucleation to cell survival in the absence of adenine. We quantify the population by deep sequencing before and after selection, and we calculate a nucleation score. You can see the effect of single amino acid substitutions in this heat map, in which blue indicates increased nucleation and orange indicates decreased nucleation, always relative to the wild type. And if we now look at the same data but stacked by position, you see there is a modular organization of the mutational effects along the sequence, and mutations that increase nucleation are enriched at the N terminus, while most mutations at the C terminus disrupt nucleation. And this modular organization agrees with all proposed structures for A-beta 42, in which the C terminus is forming the amyloid core of the fibrils, where side chain packing is very tight. Here, what you are seeing is an example of an amyloid fibril, and these are two protofilaments that are facing at each other. And if we take a slice of this fibril, we obtain this other representation here on the right. And this is a cross section, and we mapped the average effect of mutations. And you can tell that disruption of nucleation happens when we mutate the residues at the C terminus. So at the beginning of the presentation, I was telling you that there are 14 mutations that are associated to familial Alzheimer's disease. And you might be wondering, how do they score in our assay? And you can see them here highlighted in red, and they are within the top scorers or nucleators in our assay. We tested the performance of our assay in discriminating these disease variants. And the ROC curve here in red is showing you the nucleation scores accurately discriminate them. And they do it better than any other computational predictor or experimental assay done before. And so human genetics is confirming that we are tracking a biological process that is relevant to disease. And this means that we could only use nucleation scores to predict whether a mutation would cause familial Alzheimer's disease or not. And also the agreement of our data with the effect of disease mutations is telling us that familial Alzheimer's disease is very likely to be an amyloid nucleation disease, and that therefore that is the step to target. So all this stuff is part of a published story, and now I would like to move to something new. And so when we were finishing that project, we started to be very interested in other type of genetic variants, because we have to keep in mind that genetic variation between individuals is not only due to single nucleotide changes that result in single amino acid substitutions, but other type of genetic variation also exists, such as insertions and deletions. And these other type of changes can have a big impact on protein folding and function, and they can also be associated to disease, and we still know very little about them. And for example, truncated variants of the A-beta peptide have been found in pathogenic amyloid deposits and are known to aggregate faster than A-beta wild type. And here what you are seeing, this is an example of in vitro kinetic studies, classic in vitro kinetic studies that they track aggregation with thioflavin T binding. And here there's at least one truncated variant of A beta that aggregates faster than the wild type. And the good news here was that with our assay, apart from substitutions, we can also test other type of genetic variants. And so this library, in this case, apart from substitutions, it also includes insertions, single amino acid deletions, truncations from one or both sides of the peptide, and internal and larger deletions. These are the distribution of the nucleation scores. And although most of the mutations decrease nucleation, they have a negative score, we still can see that we have some variants that increase nucleation in all type of mutations. And we can also classify all these variants by their ability to nucleate, and we can split the data depending on the location. And so they can be at the N or at the C terminus of the peptide. And we see that the ones that increase nucleation here in blue are normally located at the N terminus. And we already knew this from the previous work, and it makes sense if we remember the wild type sequence in which the C terminus is composed by hydrophobic amino acids, and they are forming the amyloid core of the fibril, while the N terminus has charges and polar residues. Now let's have a look at each of these subsets, and let's start with insertions. So again, we see this modular organization of the peptide in which inserting an extra amino acid at the N terminus can sometimes increase nucleation, but inserting one residue at the C terminus disrupts nucleation. And it is interesting to see that there's a specific stretch here at the C terminus between residues 29 to 38, in which most insertions disrupt nucleation. And this is suggesting that there's a specific stretch at the C terminus that constitutes a tighter core of the nucleating sequences, and that they cannot accommodate additional side chains. We also have single amino acid deletions, which means that we are removing one amino acid in the wild type sequence at a time, and these deletions at the C terminus disrupt nucleation, but they have a more balanced effect at the N terminus. And the top nucleator in this subset is a deletion at position 22, which is actually another familiar Alzheimer's disease mutation known as the Osaka mutation. And we can delete bigger chunks of the peptide, not only single amino acids. And these are truncations from the N or the C terminus of the peptide. And we see that if we truncate the peptide from the C terminus, nucleation is disrupted completely. But we can increase nucleation if we truncate from the N terminus, as long as we keep the C terminal intact. And the top nucleator in this data set is this variant here from 28 to 42, which we call the K core, which is basically the amyloid core with just one lysine residue in front. And for the moment, just keep in mind this one variant, because we will talk about it a bit later. And it is also interesting to point out here that in our body, we have two isoforms of A beta, which are A beta 42, the one that we are studying here and we take as our wild type, and A beta 40, which is a shorter version and is missing the last two residues. A beta 42 is known to have increased toxicity and aggregation propensity compared to the shorter form. And here we have this shorter form, A beta 40, as a truncation, and it's nice to see that it also has decreased nucleation in our assay. And finally, I would like to move to those deletions that remove more than one amino acid in the wild type sequence. And we plotted all of them in this matrix, in which the x-axis is showing you the first position that we are deleting. And on the y-axis, we have the last position that we are deleting. And so, for example, this square here corresponds to a deletion that starts at position 17 and removes until position 14. And we can also split this matrix into 3 subsets: those deletions that are located at the N terminus, those that are located at the C terminus that start after position 29, and those that involve both the N and the C terminus. And once again, we see that most mutations that involve the C terminus decrease nucleation because they are disrupting this hydrophobic amyloid core. But one of the first things that we realized when we had this matrix is that those mutations that increase nucleation cluster in a very specific area. And here we could identify a hotspot of deletion effects that is between residue 16 to 25. That if deleted, nucleation strongly increases. And this is also evident with short deletions. And here I'm showing you deletions from 1 to 6 amino acids. And we see that those deletions that are missing residues in this area are the ones that increase nucleation the most. Sometime after we generated all this data, there was a paper that came out, and it was describing for the first time that there's one large and internal deletion that is associated to familial Alzheimer's disease. And this is a novel variant, is a deletion at residues from 19 to 24, and is known as the Uppsala mutation. And so once we saw this paper, we went back to our matrix and checked this one variant, and we saw that it also has increased nucleation. And so this was confirming once more that our assay is good in discriminating variants associated to disease. But what is more interesting is that this variant is falling inside our hotspot of deletion effects and is surrounded by other variants that also have increased nucleation and are currently of a non-clinical significance. And so here we think that they are also very likely to be pathogenic. And now I would like to focus on those deletions that involve the N terminus but maintain the C terminal intact. And so the result here is a variant that keeps the amyloid core but has a new N terminus. And we can check which amino acid residues we have at each position. And this matrix is showing the distance from the C terminus of these residues, and the y-axis, the amino acid that we are putting. And here, just for reference, the wild type is indicated with an asterisk. We see that amino acids have a neutral or increasing nucleation effect when they are far away from the C terminus. But in the 3 positions just before the C terminus, they can disrupt nucleation with some exceptions. And here we see that charges or glutamine or histidine can instead increase nucleation. And this is telling us that there's a specific preference for specific residues, such as positive charges, that when they are flanking the amyloid core, they can boost nucleation. And this goes a bit in line with what I was telling you before. If you remember that variant, that it was the K core 28 to 42, that it's keeping just the amyloid core fused to a positive charge. And that was the top nucleator within the truncated variants. And so at this point, we were very interested in this opposite role of positive and negative charges, because we see here that negative charges flanking the core disrupt nucleation. And so we tested individual variants, and we tested their ability to nucleate. And here we confirm that just one positive residue before the core is sufficient to boost nucleation. But so does arginine, which is the other positive residue, and that can substitute the role of the lysine. And interestingly, these charges can also be placed at the other side of the core, and although to a lower extent, they can still increase nucleation. But in contrast, negative residues decrease nucleation completely. And once we had all this data in our hands, we wanted to put it in context of the protein structure and to rationalize a bit the effect of all mutation types that we have seen here. And this is just a reminder of how does an amyloid fibril looks like. And this cartoon here represents an individual peptide within the fibril. So from most of the proposed structures for A-beta-42, the wild type would have 3 strands. These are 3 beta sheets, 3 beta strands, sorry, that are formed by hydrophobic residues, and they are facing each other thanks to these 2 connecting loops here. However, we know from our data that the minimal nucleating core is variant 28-42, which would only have 2 strands, and it keeps the positive charge in front. And we ranked all variants in our library by their ability to nucleate, and found that the top nucleators all affect and disrupt a specific area, which is this big loop connecting the 2nd and 3rd strands. And some examples of this are single deletions at positions 22 and 23, also larger and internal deletions in the hotspot area, and also substitutions in these residues. And this made us speculate that all these top nucleating mutations are bringing the 3-strand structure back to the minimal nucleating core, which is the 2nd strand structure, which can actually nucleate faster than anything else. And overall, all the different type of mutations in our library that are very different between them, they are all pointing at the same structural features and are giving us insights of a specific conformation that could be important for amyloid nucleation. And here I would like to just finish by thanking people in my lab, especially Benedetta and also Marta, and our collaborators at the CRG, Ben and Andre at Ben Lehner's lab. And thank you all for your attention, and I'm happy to take questions.
Great. Thank you, Mireia, for that fantastic presentation. We should have some time for a few questions. Just as a reminder, please feel free to post those to the Q&A or the chat, and we'll go ahead and read those. I guess while people are doing that, I have a question that I can maybe start off with. So you could mutations that prevent nucleation or aggregation generally have like a protective effect? You mentioned this for the AB40, but do you think this might happen for other similar protective variants? I thought that was a very kind of interesting example you gave.
I think we actually don't have, well, we have this clear example of A-beta-40, but we are missing what we call our negative controls that are variants that we know that can be protective, but we miss them because we don't have information about them, so we cannot really compare them to anything else. Yeah, but I think A-beta-40 is a good example of that.
Yeah, I thought it was very interesting. Okay, so we have a couple questions here. So this one's from Mike Pazin. Starts off with thanking you and saying it's a great talk. And the question is, are the gatekeeper amino acids more or less conserved than the other residues?
Sorry, can you repeat?
Yeah, are the gatekeeper amino acids more or less possibly conserved than the other residues?
I don't get what conserve means in this case.
I guess, yeah, maybe conservation across species, I would imagine, or, conserved in human populations.
I don't know. What I can only tell that I didn't explain it here is that we define some gatekeeper residues in this project. Sorry, let me put the pointer here. So we define some gatekeeper residues in here, in which are the ones that most mutations increase nucleation, which means that you cannot really mutate them. And most of them were negative residues. And we know that negative residues in other amyloid proteins can also act as gatekeeper residues. So I don't know if this can answer the question, but at least.
So, I think, Laura, maybe can you unmute Mike to, I think he's here, we can have him ask.
Yeah, Mike, I think you're permitted to talk now from the attendees.
Thanks, Laura and Diego. Yeah,
Sorry,
I was
thank
asking
you.
about sequence conservation across species or constraint within human. Is there less variation or more variation at these residues that are critical for determining aggregation rate?
Yeah, so I think in general, A beta is not functional by itself. So conservation does not really, it's not really important. And so we don't know if they are really conserved. Like there's no evolution pressure in this sense.
Thanks.
Okay.
Yeah, so we have another question here from Sunak Sahu. They also say, great talk. How do the nucleation scores correlate between the replicates? And if you see variants, how do you tackle that during statistical modeling to classify variants?
Yeah, so I haven't shown this, but yes, in this in this first paper, we had really good correlation between replicates. I think we had 3 or 4 biological replicates, and also we know that there's also good correlation between technical replicates. And what we also validate is that variants behave in the same way or have or correlate, their scores correlate when we test them in small scale, which means that we take individual variants and we validate their score in the large scale experiments.
Okay, well, that looks like all of the questions that we have. So we're going to move on now to our second talk. So thank you, Mireia, and also especially the fact that it's midnight there where she's presenting. We all appreciate you staying up to do this. So thank you for your talk.